This is a spotter in general microbiology. This is seeds filter used to sterilize material that get destroyed in the autoclave. The disc that is used for sterilization is asbestos filters. Seeds filter used for sterilizing sugar solution, antibiotic solution, and sugar solutions. The next spotter is Dreyer's tube, and it is used for doing Vidal test. Vidal test is a slide agglutination test done for the diagnosis of enteric fever. This is sterile swab, which is usually sterilized by hot air oven if you prepare domestically. If you buy from commercial this thing, either it is sterilized by gamma radiation. This is low bond comparator. Optimum pH is 7.4. The organism which grown in alkaline medium is Vibrio cholera. Acidic medium is fungi. Optimum pH is 7.2 to 7.4. This is a syringe. Again, it is sterilized by ETO, ethylene oxide or gamma radiation. It is used. It is packed and sterilized, and it is used for picking up. It is used for taking the blood for blood culture. Hot air oven. Dry heat is used. Temperature identified. Temperature employed is 180 degrees for 1 hour, 160 degrees for half an hour. So all instruments, glasswares, swabs, antibiotic solution, swabs, oils, instruments are sterilized in this. Glasswares, very important. Glasswares are sterilized. This is autoclave. Temperature employed is 121 degrees, 15 pounds pressure for 21 minutes. So the temperature employed on boiling water, the temperature increases, and it is widely used in hospital as well as microbiology practice. And this is inspicitor, 80 degrees centigrade for 3 successive days. First day vegetative forms get killed. Second day all vegetative forms are transformed to spores. If spores are transformed to vegetative form, they get killed. It's used for sterilization of LJ medium and Loeffler serum slope. This is bacterial inoculating loop sterilized by red hot sterilization. It is used for picking up colonies and inoculating into the liquid media. This is sterile. This is Pasteur pipette, and it is commonly used for transfer of sterile liquid medium from one tube to another tube. And it is candle filters. This is usually commonly employed in the house for sterilization of water. And this is kept as a spotter. And this is sintered glass filter. Again, the filters which are used for sterilization of antibiotic solution, sugar mediums, and serums, which are destroyed by autoclave. And this is brain heart infusion broth used for taking the blood culture. Blood agar. Blood agar is nutrient agar plus 7% sheep RBCs. It's an enriched as well as differential media. It is used for growing streptococci. Alpha, beta, and gamma streptococci. Alpha is partial zone of hemolysis, beta is complete zone of hemolysis, gamma is non-hemolytic streptococci. Heated blood agar is called chocolate agar. It is also an enriched medium, and it's used for growing Neisseria and Haemophilus organisms. This is TCBS, thiosulfate citrate bile salt sucrose agar, used for growing Vibrio cholerae. It ferments sucrose, colonies are yellow in color. LJ medium contains mineral salt, asparagine, glycerol, and beaten egg, sterilized by inspisation, and it is dispensed in screw cap bottle. It is used for growing Mycobacterium tuberculosis. This is CBME BATCH dish diffusion method for doing antibiotic sensitivity. And please remember, the zone of clearance is called as zone of inhibition. This is McConkey's medium. Please mnemonic, you remember: plant, peptone, lactose, agar, neutral red, and taracolate. Differentiate between lactose fermenters. Lactose fermenters look pink in color, not lactose fermenters look paleless or colorless. This is peptone water, usually used for growing the organism for doing antibiotic sensitivity. When you add nutrient to it, protein to it, it becomes nutrient broth. This is Castrinata biphasic medium. It contains solid as well as liquid medium. Solid medium as slant, liquid medium as broth. It is used for usually going brucellosis. Robertson cooked meat medium, minced cooked, it is added. Paraffin layer is added to create anaerobiasis, used for growing anaerobic organisms. Now coming to the immunoglobulin, a pair of light chain, pair of heavy chain held together by disulfide bond. Abundant antibiotic is the only immunoglobulin passes the placental barrier. This is IgM, mainly pentamer. It has got 10 antigen combining sites. It is predominantly intravascular in distribution. This is IgA, secretory antibody present in all secretion in the body. It has got 4 antigen combining sites. And VDRL, venereal disease research laboratory test, done for the diagnosis of syphilis. This is tuberculin saline for use for doing Mantoux test, 0.10 ppd or 0.1 ml of purified protein derivative or whole tuberculin is injected in the volar surface of arm. After 78 hours in duration is measured. This is MMR vaccine, usually given in 12th month. Mumps, measles, and rubella vaccine. Hepatitis B vaccine is one of the beautiful vaccines, DNA recombinant technology, and used for given in 3 doses: 0, 1 month, and 6 months. Booster every 5 years. DPT, diphtheria, tetanus toxoids, and pertussis vaccine, given in 6th week, 10th week, and 14th week, followed by 18th month and 6 years, 12 years, and 18 years. LHL precipitation test done for the detection of toxin of Corynebacterium diphtheria. Naegler's test, this is done for Clostridium elchi to detect alpha toxin. Mantoux test, again, it is a delayed hypersensitivity reaction done for the susceptibility, skin sensitive test for the tuberculosis. Systemic bacteriology, this is gram-positive cocci clusters, staphylococci. This is gram-positive cocci in chain, streptococci. This is a long chain, long, slender, slightly curved acid phosphate tuberculosis. This is a McConkey's medium showing pink colored colonies, lactose fermenter. This is pale colored colonies, Salmonella shigella proteus. Examples of non-lactose fermenters. This is showing blood agar with beta-hemolysis, usually staphylococci. Cement citrate agar. Cozer is liquid, cement is solid. Growth or change in the color is positive. Triple sugar iron agar contains 3 sugars: glucose, lactose, sucrose. Ferrous ammonium sulfate to detect H2S, and which detects H2S formation. You see acid but and alkaline slide, and there's only 2 sugars are glucose is not fermented. The urease test contains peptone, agar, neutral red, phenol red, and urea. Highly urease positive organism protease. Virology, the various routes of inoculation of egg, corentonic membrane, corentonic cavity, yolk sac. These are various The vaccines that are prepared in the chicken or anti-rabies vaccine, and rickettsia and chlamydia are grown in yolk sac. This is an agribody seen in rabies. Microtiter plate used for um, hemagglutination inhibition test. This is Ebola virus. Next is transport media, viral transport media, and is used for example, in COVID time, used for transport of nasal medium. This is cycle showing RT-PCR. Can look for the CT cycles, various CT cycles. Parasitology. This is a tapeworm, dioecious. This is a roundworm, which is male and female roundworms. This is tapeworm, hookworm. NIH swab, National Institute of Health swab, used for taking the rectal swab, anal swab or rectal swab from the perianal region in Enterobius vermicularis. This is a of non-bile stained of hookworm. This is a whipworm, bile stained with mucus plug at both end. This is a gametocyte of Plasmodium falciparum. This is Entamoeba histolytica showing quadrinucleate cysts and various other cysts. This is Trichomonas vaginalis, one of the sexually transmitted disease, parasitic disease. This is a of Taenia solium. Mycology. This is Sebrodextrosacar, pH is 5.4. This is slide culture. B glass slide used, and 2 to 4 centimeters Sebrodextrosacar is put with colonies. Can appreciate the morphology. So Aspergillus on Niger. It looks like black. It's Niger. And Rhizopus species. Can see this aerial mycelia. This is Candida. Candida and Cryptococcus resemble each other. This is Sebrodextrosacar. Cryptococcus, you have to differentiate by doing urease test and jump to formation. That is Penicillium on Sebrodextrosacar. And this is Rhinosporidium severi. It's not been cultivated in the laboratory. This is Penicillium species. It looks like a broom. A Rhizopus, which has got roots, rhizoids. This is Candida species. It's pseudohyphilium. This is Cryptococcus, a negative nigrosine or Indian population. Thank you very much.